So I wanted to share with you today what my company does, and then just sort of more broadly about kind of this new field of science and medicine that's centered around the microbiome. So if you were an emperor or a high official in ancient China, it all starts in ancient China, of course, and you got sick, you would oftentimes be given what was called the golden soup. And the recipe for the golden soup is you take stool from a newborn infant and you mix it with hot boiling water, and then you drink it. And there is documentation as early as 4th century China from these physicians saying that they were able to cure people of diseases using this golden soup. So what they were doing then that we now understand was what's known as a microbiome transplant. So the microbiome is all the bacteria and viruses and fungi that live inside and on us, and it turns out that they play a really important role in our health as well as in disease. And so I co-founded Pendulum Therapeutics about 6 years ago, and the whole goal of the company is we exist to make millions healthier through microbiome-targeted medical probiotics. I was initially drawn to this space for a very personal reason, and that is my daughter. So this is Annabella Cutcliffe. She was born almost 8 weeks prematurely, and when you have a baby that's born that early, you basically get to see the baby for a few seconds and then they're whisked off to the ICN. And Annabella spent the first 4 weeks of her life looking like that. One of the other things that happens when you're that premature is that you're given multiple doses of antibiotics, not because you have an infection, but because you're so fragile that they want to prevent you from having an infection. And one of the things that I've come to notice about Annabella as she's gotten older, she's 12 now, is that she has certain food sensitivities that nobody else in our family has. And as I started to learn more about the microbiome, I started to realize that maybe this early start to life was particularly problematic for her because antibiotics is one of the most potent ways to disrupt a microbiome. So what happens when you take antibiotics are a few things. So this is sort of a picture of your gut microbiome here. In the beginning, you have this really nice ecosystem of all these microbes in your gut. When you take an antibiotic, 3 things happen. The first thing that happens is that almost all of the strains that are in your gut get entirely decimated by that antibiotic. The second thing that happens is you can have something that is really at a low level, but now that there's no competition, it can start to propagate unchecked. If you've ever heard of the C. diff infection, that's really what this is. Many of us carry C. diff, Clostridium difficile, a strain in our guts, but for some people when they take antibiotics, it kills off all of these different strains, but their C. diff is still able to live. And then when it starts to replicate like this and get to really high levels, it'll make you really sick. Ultimately, it's fatal. The third thing that happens is eventually, over time, your microbiome will reconstitute itself, but oftentimes what it's reconstituted with is actually not the same thing as what you started with. So for decades, farmers have actually been giving their cattle antibiotics, not because they were trying to change their microbiome, but because they observed that when they gave their cattle antibiotics systematically, they became more obese. Well, that's not just true in cattle, it's also true in people. So in a study that was done across 12,000 children, they found that children who were systematically on antibiotics before 6 months of age were also systematically more obese as they went into their teenage and adulthood years. So then there was this sort of really cool seminal study that was done, it's been replicated lots of times, where people were trying to understand what could be the role of the microbiome in metabolism, and can the microbiome actually change your metabolism? So this was an animal study in which they took the microbiome from this obese woman, they put it into this thin mouse, and the thin mouse became obese. They then took the microbiome from this thin woman and put it into an obese mouse, and they saw that obese mouse become thin. Then they took the microbiome from the thin mouse that got it from the thin woman and put it into the obese mouse that got it from the obese woman, and they were able to get that obese mouse to now become thin again. And the really cool thing about this study is that these 2 women are identical twins. So this whole thing is just microbiome manipulation changing metabolism. So if you were to look at all of the publications that have happened in a simple PubMed search, you would see that over the last decade there's been a huge amount of information that's been uncovered around the microbiome. So in blue here are publications on, if you just type in microbiome, and then in red is if you type in microbiome and metabolism, and you can see that there is growing work around microbiome and metabolism. So things like probiotics and yogurts have actually been on the shelves for decades, so you might be wondering, what's with the 0 things happening before 2007? So this is actually really cool for all of us who live in the Bay Area and Silicon Valley, because it is DNA sequencing technologies which have become affordable and actually usable, that were all developed here in Silicon Valley, that became available around the mid-2000s. And because DNA sequencing became available and affordable, we were now able to survey our microbiome. Your microbiome is comprised of all of these different bacteria that have about 100 to 1,000-fold more genes than the human genome. And so in order to really survey the microbiome, you have to have really hard-hitting DNA sequencing. And so DNA sequencing technologies are what have enabled microbiome to even become a science and a medicine. So after all of this research, we have our first microbiome therapeutic. That is what you think it is. And that is also what you think it is. These are called fecal microbiome transplants. And essentially, we are no further along today than we were in 4th century China. So people are using these fecal transplants in order to resolve disease. And one of the things that you'll find is that it's actually been extremely effective. For that C. diff infection that I talked about earlier, the protocol is actually to take antibiotics in order to kill off that strain. And that has about a 30% success rate. These fecal microbiome transplants have about a 97% success rate. And remember, success and failure is life and death here. And so you have people volunteering to actually go get these fecal microbiome transplants because they know that this is what's going to be able to rescue them. So at our company, we thought we could certainly do better, insert all your favorite jokes here, than this shitty therapeutic. We can do better than this shit. And the idea was, can we figure out if the composite, the fecal microbiome transplant, can confer benefit? What are the specific components that are actually conferring that benefit? And so if you could figure out what are the strains of the small molecules or the prebiotics that are actually doing the work, you could then create an intervention that had specificity, manufacturability, and ultimately FDA approval. And while fecal microbiome transplants are a thing of today, they are not going to be a thing of the future. As people start developing these kind of specific interventions, those are all going to go by the wayside because frankly, at the core of them, they're unsafe. You really don't know what you're getting. You need something that you can manufacture. So again, when we started the company, I actually started with 2 people, and we worked in a DNA sequencing instrument company. So this is how we really understood how to use DNA sequencing in the application of this field. And when we first started the company, people were creating several of these consumer diagnostic companies around the microbiome and able to offer sequencing for things like, less than $100. And they were really getting what is called genus level resolution. And what we were doing was actually getting strain level resolution. And this is the kind of resolution that you get when you spend thousands of dollars per sample. So we've never been a consumer-based company. You can't tell people to spend thousands of dollars just to get some microbiome sequencing. But we really believe that this is the kind of data and insight that you need if you want to develop diagnostics and interventions in this space. And so when you think about genus versus strain level resolution, this is really trying to remember 7th grade biology. There's phyla and family and genus, but the strain level resolution is thousands of dollars per sample if you really want to understand it. Most of the consumer-based companies are getting genus level resolution. So we started the company with this premise that we can create this discovery platform which integrates lots of different data types in order to understand how to identify interventions. And I'm just going to kind of speed through this really quickly, but just for some terminology. So the first is microbial classification. So this is who are the players. So if you ever hear people talking about 16S sequencing, that's what they're getting. They're talking about what are the different microbes, what's the catalog of them. We wanted to more deeply understand what are the genetic capabilities, and that's where you get the full genome sequencing. So when people talk about shotgun sequencing, metagenomic sequencing, assembly, that's all referring to actually getting down to what are all the genes, not just who are the players, but what are they capable of doing. Once you know who the players are and what they're capable of doing, then you really want to test actually what are they doing. So you're looking at what are the enzymes, what are the biochemical reactions, what are they capable of performing in terms of metabolism. And then lastly, most importantly, and probably most difficult to obtain, is how does the microbiome interface with a particular host? So how does it interface with your immune system, your inflammatory system, and how is it different from person to person? And that involves actually needing to get microbiome interventions into people. And so that has been the name of our game since we began, is how do we get interventions into people as quickly as possible and try to understand how we are affecting health. So the microbiome really represents this, I think, really unique opportunity, which is that if you're using science, you can sort of meld the precision of science with the harmony of nature. And what I mean by that is that we believe that you can create products in this space that are as efficacious as small molecule drugs, but they're as safe as over-the-counter probiotics. They can restore the body's natural capabilities. You can create natural, non-GMO organic interventions, and kind of most importantly, you can create lifestyle products that people can love and feel good about. I was going to talk to you about some of the work that we're doing in diabetes, and diabetes is a great example of a disease where there are tons of drugs that have actually great efficacy. But one of the biggest problems with diabetes drugs is actually compliance. So insulin is a drug that you have to inject multiple times a day. GLP-1s are products that you have to inject multiple times a day. I used to work in pharma, and when you're developing a pharmaceutical drug, you don't think of the end user or you don't think of the consumer. You just think about, how do I develop something that's efficacious? You're just checking the first box. But when you talk about developing products that are for people that you want them to use, and you can create a lifestyle product that people really enjoy using, if you have efficacy and compliance, that's actually how you can really change health. And that's really the opportunity that we see and that we're going after. So now I'm going to fast forward like 4 years of work into this single slide. We have a product, and it is a pill that has a bunch of different microbes in it, so bacterial strains in it, and the prebiotics that feed those strains. These are entirely novel. We discovered them through our sequencing as well as analysis in the lab, and I'm just going to briefly tell you how it works. So this pill has two functions. One is in the metabolism of fiber. So we all know a high fiber diet is really good for us. We're also supposed to be eating lots of fruits and vegetables. But it turns out that we don't encode for anything that can actually metabolize that fiber. It's all metabolized by your gut microbiome. So if you don't have microbes that metabolize fiber, that fiber is literally going right through you. And one of the reasons why fiber is really good for you is because once it's metabolized by your gut microbiome, it creates these small molecules called butyrate. Butyrate binds to G protein-coupled receptors, which results in GLP-1 secretion, and that leads to improved diabetes through lowering blood glucose spikes as well as A1C. So dietary fibers, this market is a multi-billion-dollar market. It's one of the only products that if you have enough fiber in your product, you can actually make health claims around it. Downstream of this, GLP-1, insulin, these are all a multi-billion dollars worth of products for diabetes too. But there has never been a product that was the microbiome product that actually did this metabolism of fiber into butyrate, because no one's ever known what it was. And that is at the core of what our product does. The second function that it has is really around your gut lining. So if you ever hear people talk about leaky gut or mucin layers, really what they're referring to is that you've got this great gut microbiome that's doing all these things, producing all these small molecules, but if your gut lining is misregulated, you can have essentially these small molecules now escaping the gut where they're supposed to be and getting into your bloodstream where they can cause heightened inflammatory immune responses. And so the goal of our product is to first metabolize fiber into butyrate, and second, to keep that butyrate where it's supposed to be by enhancing the gut lining regulation. So, just to give you a feel for some of the kind of hardcore in vitro science that we do, I'm a biochemist by training, and so for me this part is actually the exciting part, but I can appreciate it's not that exciting for other people. Essentially, we have the ability to profile all of our microbes. So these are just 3 different kinds of things we profile in-house. So on the left-hand side here is the growth. So this blue line is the growth of a strain. This orange line is the pH. We're creating butyrate, butyric acid. So as it gets created, you expect the pH to go down. We can then measure the actual production of these short-chain fatty acids. This is all in test tubes in the lab, but we can also measure it in people once we get the product into people. And then lastly, we can measure stability of the product in different conditions. And so you're trying to mimic what are different conditions and how stable are each of these different strains in those conditions. After we did that characterization, we did this animal study. This is a preclinical study in these mice, which are DIO mice. And these mice are wild type genetically, but they become obese and diabetic the same way we do. They feed them a high-fat, high-sugar diet. And we've basically done our whole development as though we were developing a pharmaceutical drug. So this is a study that has 4 arms. It has a negative control, a positive control, and then we were testing 2 different formulations. One that only had the strains, and that one had the strains and the prebiotics. So the probiotics, when you hear this term, it's actually the bacterial strains. Prebiotics are the food for probiotics. And so the idea here was, I always like to think, like, if I were dropped off in the middle of a deserted island, would I rather have a basket of sandwiches or be by myself? And so the idea of giving them the prebiotic means that you're giving them the food to help them once they revive in the gut. And so we gave them to these mice, and we were measuring their oral glucose tolerance test. And so I'll just take you through this for a second, because this is one of the hallmark gold standard tests for diabetes. If any of you have ever potentially had gestational diabetes, or when you're pregnant you are at risk for that, you will have undergone this lovely test, in which what you're trying to understand is how does somebody metabolize sugar. And so you take their blood sugar levels at baseline, and then at time 0 you take this big bolus of sugar, and then what you're looking for is what happens to the blood glucose level. So if you have diabetes, you get these really big, everybody gets a spike after you have sugar, you get a spike and then it goes down. But for people with diabetes, they get a really large spike, and then it takes a while for that glucose to be metabolized. And that's really the heart of diabetes, is the inability to metabolize sugar properly. And so when we gave our product, we see a reduction in this, which is the red line here, by 10%. And that 10% is referring to the area under the curve. So the way that you compare these is you're actually calculating the area under the curve, and you're saying, all right, there is an improvement by 10% in the way that this animal is metabolizing glucose. So then we moved on into people. And so this was a really small pilot study that we did. There's 6 people, 3 of them are healthy, 3 of them have prediabetes. And we did this as a placebo-controlled, double-blinded, randomized crossover design. And we used continuous glucose monitors and meal tolerance tests. So a continuous glucose monitor is something that anybody here can buy. You basically attach it to your arm, and it measures your glucose in real time. And so we used this, and essentially each person got randomized to either get a placebo or treatment first, and then they got the opposite, either the treatment or placebo. And we gave them this same glucose tolerance test at the beginning and end of each period. And what we're trying to understand is, do they look different when they're on the treatment at the beginning and the end versus on placebo at the beginning and the end? And so just to give you a feel, this is like you just get massive, massive amounts of data with continuous glucose monitors. Each of these colors represents a different person in the study, and each of those spikes is anytime somebody's blood glucose went up. So this happens whenever you eat something with sugar, drink something with sugar. Actually, when you exercise, your body starts releasing all this glucose. So there's all these different things in which that you're doing, knowingly or unknowingly, to cause these sugar spikes. And so where you hear the most about continuous glucose monitors is actually for kids with type 1 diabetes, where they really need this real-time monitoring in order to know how much insulin to give them that's appropriate. But this is becoming a technology that is getting more and more widely used. So I told you that we had 3 healthy people and 3 people with prediabetes. This is the glucose tolerance test curves for each of those people here. And I also told you that people with prediabetes are unable to manage sugar. So I'm going to show you the labels of these, but I bet that everybody here can guess who are the 3 people that have prediabetes and who are the 3 people that are healthy. It's 1, 2, and 4. So these 3 people have prediabetes, and you can see that they get this sugar spike when you give them their, this glucose spike when you give them the sugar bolus. So we measured, again, the same thing where you're measuring the area under the curve and you're looking at people on placebo versus product. And what we found is that the 3 people with prediabetes, when they were on the formulation compared to placebo, we saw a reduction in their area under the curve by 24%, 57%, and 74%. You just remember these are small numbers, and so, but it was really compelling for us to see that and to feel like we might have something that has impact here. We didn't see a change for the healthy individuals, but that's because they don't have this problem with the way that they respond to glucose. So then we went on and did this larger study. So this is a 12-week double-blinded placebo-controlled trial, and it's in patients who have type 2 diabetes, and they're at an early stage of diabetes. There are 3 arms here. There's a placebo arm, and then there's 2 different formulations. And here we were testing whether you need both the fiber metabolism and the gut regulation, or if you could just get away with the fiber metabolism on its own. This was 76 people across 6 sites in the US, and we were looking again at their area under the curve, but then also at their A1C. And so A1C is the other diagnostic marker for type 2 diabetes. And so, again, this was like a 2-year study. So this is it's always great when 2 years of work can be summarized in one picture. So this is the 2 key pieces of data. Essentially, the placebo is in yellow. The light green is the formulation that only had the ability to metabolize fiber. And the dark green was the formulation that could metabolize fiber and keep your gut lining regulated. And what we found was that you can see some trending for the one that only did the metabolism of fiber, but actually when you have both of these functions, you get a statistically significant result that shows that you lower A1C by 0.6 and you lower AUC by 24%. So these are both statistically as well as clinically significant. And just to give you a feel for what these numbers mean in a clinical context, on the right-hand side here is A1C. So you have diabetes, and just in case anybody's interested in what A1C is, this is a measurement of your blood cells that have glucose attached. So it's actually hemoglobin A1C. So everybody, you have red blood cells, and when you have too much glucose kind of floating around in your blood, your body does this thing where it attaches sugar to your blood cells. And so people who have type 2 diabetes have elevated numbers of these red blood cells that have sugars attached to them because they've got all this free-floating sugar. So if you have an A1C of 6.5% or higher, you have type 2 diabetes. If you're between 5.7 and 6.4, you have prediabetes. And if you're lower than 5.7, you're normal. And so you can see that with a drop of 0.6%, that can actually mean the difference between having diabetes and not having diabetes, or having prediabetes and not having prediabetes. And when you look at the efficacy of all the small molecule drugs, or the key small molecule drugs that are out there, because there's like 40, you can see that we're right in range with the kind of efficacy that that drugs have. But our product is actually a probiotic. It's really these naturally occurring strains and the prebiotics that feed them, whereas all the rest of these are drugs. And then at the far end there, GLP-1s and insulin are actually injectable drugs, so not an oral medication anymore. And then I think one of the most important parts is the side effect part here. So one of the reasons why people are not compliant is also because of side effects that they experience or could experience through their drugs. And so one of the advantages to probiotics is also not having some of these terrible side effects. So for example, sulfonylureas, even though you have reduction in your A1C, you also are at an increased risk of cardiovascular disease. There's also things like hypoglycemia and what are some of the other, like, kind of, yeah, well, you can read the list of all the terrible things that you don't want to experience as a side effect of your thing that you're taking. Yes?
Just curious if this was compared to changing a diet in a particular way, what would be the difference in impact, and what would you expect to see in terms of reduction in movement between pre-diabetic and normal, et cetera?
Yeah, I mean, diabetes is one of those diseases that has been earmarked as a lifestyle disease. So usually there's obesity, then prediabetes, then diabetes. And so there have been a lot of studies that show that if you dieted and exercise, that you can also, I mean, essentially you can reverse this too. You can lower A1C. It doesn't work for everybody. And so kind of one of the missing components has always been like, well, what if they don't have the right microbiome, but they are changing their diet? But certainly there has massive amounts of data showing that if someone, for most people, if they were to diet and exercise, that they can reduce their A1C. The problem is that we've been talking about that for, 50 plus years, and it has continued to, what?
They'd rather take a pill than change their...
It's true, and this is not intended to replace that. So certainly everybody should diet and exercise properly, but this is really just to help people get the microbes that they otherwise don't have. And to be honest, I mean, I'm no anthropologist or sociologist, but it feels like we've had the same message of diet and exercise, and it's not working. So we need to come up with other solutions. We need to help people to be able to achieve their health goals. Yes.
So,
oh.
Once you get the microbiomes in your system and you've taken this for a while, do you have to continue taking the pill, or have you just corrected yourself by taking this?
So it depends, actually. We'll come back to this diet point, which is that for most people, the reason why they don't have these microbes, the hypothesis, is that they're not eating a diet that allows these microbes to survive. And so if you had a high fiber diet, you would be giving them the food. So in the absence of changing your diet or exercise or lifestyle, you probably have to keep taking these strains and reconstituting. In our clinical trial, when we looked at people, and that's the 76 people, so it's less than 100 people, but when we looked at these patients, we did this study for 12 weeks, so 3 months of taking the product daily, and then we did a 4-week washout period at the end. And after the 4 weeks, we went and looked at their gut microbiomes, and about 15 to 20% of people actually were able to sustain the strains after 4 weeks of not taking the pills. So we don't know what it is about them or about their microbiomes that enabled them to keep these strains around, but there is something interesting there about potentially not having to keep taking them over and over again.
Oh yeah, my question was pretty much a little bit like that, but you're hearing a lot about that now. We're taking all this kombucha and probiotics, and people are starting to say, well, wait a minute, how do you know you can keep these things alive in your gut? And isn't there something other than what you're eating that makes, like whether, I don't know if it's pH or whatever it is, but makes it, your gut, one person's gut hospitable to these strains versus not, another person's gut?
Definitely. Well, it's not just things like pH and all the other stuff you're eating, but it's also lifestyle. So we know that things like stress and even day and night, these can change people's microbiomes. You go on an antibiotic, you try Thai food for the first time. These are all things that totally disrupt your microbiome. And so it's the advantage of the microbiome is that it is mutable. You can change it. The disadvantage is that you can change it. And so it's a constant moving target. And so I think this is some of the stuff that needs to be unraveled. And I also think when we talk about things like kombucha or some of the probiotics that are out there now that are not backed by any real science or medicine, you run this risk of really not understanding what are you delivering and what is the impact of that. And so I think the really important thing that I really want to highlight here with you guys is that what we've created is the world's first and only medical probiotic. And I think this is the beginning of a future wave. In the next 5 to 10 years, I think you'll start to see things like this appear where it's not your same old probiotics that are on the shelves right now. And just so you know, probiotics that are on the shelves right now are comprised of the same 15 to 20 strains that have been around since the 1970s. And it's been a bunch of beautiful marketing innovation, but really not any ingredient innovation. And what the microbiome space offers is the opportunity to create novel ingredients, novel products, and then to actually create medical data behind them. So the question to ask is, what is your probiotic doing for you? What is it trying to help you do? How does it work? And what's the evidence that it's actually doing that? And we are really trying to elevate this whole field by saying, this is for people with type 2 diabetes. It has clinical data behind it. We're going to continue to do more clinical trials, but it's a different kind of probiotic. Yes?
So you're saying there's no science behind the kombucha or sauerkraut or any of these fermented foods or any of this? There's no science behind this?
Yeah, I don't want to be rude, but I mean, not really, no. I mean, what you're looking for is sort of peer-reviewed published data, which would have a placebo-controlled clinical trial behind it, and they really don't. I'm not saying that they don't work.
There's a clinical trial evidence behind it, when there's science behind it, but it's not like a clinical trial.
Exactly, thank you. Yeah, but I don't want to say that they're not working for people. There are definitely health benefits for them. It's just that they haven't been approached with the same rigor that you would approach developing an intervention. Sorry, yes?
So how do you know if you have a healthy biome or not? Is that self-evident if you feel fine, or is that something people test for?
It's a great question. The million-dollar question is, what is a healthy biome? And I think the answer is we don't know what a healthy biome is. I think the way to think about a microbiome, though, is that it's comprised of all these different functions, metabolic functions. And so if you're deficient in a particular function or your ecosystem is somehow imbalanced in a way that you can't do certain things, metabolize certain foods, then what you're really trying to do is to gain that function back. But a healthy microbiome is yet to be defined. And in fact, you can take a bunch of ostensibly healthy people and look at their microbiome, so that 16S sequencing and even the genomic sequencing, and everybody looks completely different from each other. But when you start to get down to these core functions, that's where you start to see trends. And I didn't mention this, but actually the reason why we went after these particular strains and these particular pathways is because we did a twin study, similar to that twin study that I talked to you guys about earlier, where we looked for twins that were discordant, where one twin was healthy and the other twin had type 2 diabetes, and we looked for differences in their microbiome. And what we found was that the healthy twins had vast abundances of these strains and functionalities, and the sick twin actually had none of them that were able to do this. And then there were also 2 large studies that were done, one in China and then one in the Netherlands, I think, which also showed differences between people with type 2 diabetes and people who are healthy. And so the idea is that it's probably more about the functions than it is about the microbes, but it hasn't really been figured out yet.
Is there any application for this kind of thing to things like ADHD and other neurological dysfunctions? Are you working on that end of it or just the diabetes side?
So I believe that there is huge opportunity when we talk about gut-brain disorders, and I think that this is sort of a growing opportunity. And actually, we have a second product which is early in development stage that we've been developing primarily with Johns Hopkins, and we've actually been looking at pain, particularly associated with IBS. But the thing that they just actually literally this past week shared with us, which is really cool, is that in the preclinical studies that they did, they found that when they gave our product versus placebo, that they were able to see neurogenesis in the gut. So I don't know if anybody knows this, I just learned this. You have neurons in your brain and you have neurons in your gut. And the neurons in your gut are able to produce all the same neurotransmitters, so dopamine, serotonin, all these things. And there's actually a path for those small molecules to get from your gut to your brain called the vagus nerve. And so the new hypotheses that are coming out are saying, hey, Parkinson's and Alzheimer's, where we've only been looking at the brain, and actually I used to work in Parkinson's disease, and we were only looking at the brain and these Lewy bodies, that actually you can, before anything shows up in the brain, you can already start to see that same phenotype happening in the gut. So perhaps it is the gut neurons that are actually starting the problem. And the big difference between your gut neurons and your brain, the neurons in your head, are that these turn over. These don't. What you got is what you got, and you're killing them daily with alcohol and whatever else. But these turn over. And so there's a real opportunity to intervene here where the signaling pathways can actually be altered. Yes.
So there's been a lot of discussion about transplants. Transplants. How do they actually figure out, if you don't know what a healthy gut or a biome really looks like, how the heck can they do these transplants overseas and they're allowed in the United States, the FDA will approve it? Is that an area that you look at all when you're going into healthy syndromes?
Well, I look at fecal microbiome transplants as evidence that there's something in that microbiome that could be beneficial. I think, I was sort of saying earlier that fecal microbiome transplants are something that people do today, but I think in the near future there will be no such thing because people will start to target things. It is. So I think, though, there are diseases where people, like I was talking about the C. diff infection, where it's fatal, right? And so in this case, how can you deny somebody that, even though you realize there's a risk associated with it?
And about 85%
Success rate.
success rate
Yep.
with C. diff patients that do have that, and it's crazy. I mean, they don't understand it, but, yeah, they're just looking for a way to make money off of it. But do you have a lot in the US? Well, they only if it's C. diff, it's that extreme.
So, I mean, I would say that the way that that's working is, I think, relatively straightforward, frankly, compared to trying to find specific functions. So you have people who take antibiotics, right? And now there's no competition, so they have C. diff, which is now taking over their gut and making them sick. The fecal microbiome transplant is simply reintroducing that ecosystem. And so all you're doing is you're inundating the person's microbiome with new strains that are going to outcompete that Clostridium difficile, and that's how you're wiping it out. So sort of like the thugs approach to solving a problem, right? Just like throw the kitchen sink at it, and you're basically outcompeting that strain without having to go after a specific function. But definitely it's highly efficacious. Yes?
So about IBS, is there anything you recommend in your product sphere now, or is that under development and still not available?
It is under development. We do have actually a hidden friends and family site because I'm happy to share that. Yeah, and we have people who've been on it that have been on it. We've started getting initial. So let me take one step back. All of these strains go through a GRAS certification. That means generally recognized as safe. And so we have to create a dossier of data showing that they're safe. As soon as you get that, that's when you can start putting them into people. So at that time, we kind of created this beta site where we said, well, let's see if it helps with people. At the same time, we were doing these preclinical trials with Johns Hopkins. And we released that site about a year ago, and we have people who have bought consistently for all of that time and taken these pills, some of them multiple pills a day. And so, yeah, I'm happy to share that out. We're not really sure what to do with it at this point because we don't have clinical data behind it, but we definitely have individuals saying they're benefiting from it.
So how can I get that?
Okay, I will give you my information after this.
Maybe you can share it privately with me, and I can share it with people that actually email me too. 'Cause they-
Sure.
Great.
Yeah, because I think there's a lot of people that have
No,
got
that's
issues.
a
It's
big
huge.
issue.
It is. I mean, there's a reasonable number of probiotics out there right now targeting, just to be totally honest, there are a bunch of probiotics out there that target IBS. This is not any of the strains that you would find in those, and it is targeting a specific pathway, but it's, if it works, it works. So, yeah. Yeah.
Do you plan to do any research into depression? So I've read some studies that as you were mentioning before, that now they believe that depression actually begins in your gut. Would that be something that you would take on?
I think depression is really interesting. We are still a startup company of less than 50 people, so we have to stay focused. And really, the first thing is around metabolic disease. The second thing is around IBS. This idea, though, of neurogenesis, if the truth is that the product that was talking about for IBS does actually stimulate neurogenesis in the gut, you can imagine there's a bunch of other things that it might be helping with. And so if that's really true and that repeats, then we would be looking at things like anxiety and depression. We would actually look at Parkinson's and Alzheimer's and see whether there was anything there. And then the one that I'm really interested in is autism. So there are a lot of studies showing that kids with autism have different microbiomes from their healthy siblings. And then there is also some evidence to show that certain kids with autism, when they change their diet, that you can start to see some resolution of disease. And so to me, that kind of points to potential microbiome opportunity. And of course, the all-natural profile here would be really beneficial for kids. Yeah. Yes.
I'm just wondering about inflammation in general, since that's the root of many disease pathways. And so are you viewing that as a category of just, inflammatory diseases? Or is that like a totally different?
Yeah, well, I think that the microbiome is very much tied to inflammation, inflammatory diseases. How it's tied to it is, I think, is very complicated. So in our clinical trial, we measured panels and panels of inflammatory and immune markers, and there didn't seem to be an obvious correlation between responders in terms of the diabetes outcomes and inflammation. And so we're really trying to understand, and this is probably going to take a deeper dive into the person and their genetic makeup and really try to pull that data together. But it's definitely, I think, a huge opportunity.
So back to your very first slide, is there work being done with this around obesity?
Well, actually, I was hoping that we were making a skinny pill, frankly. Yes, I was motivated by my daughter, but also motivated by wanting to eat whatever I want. So, yes, unfortunately, we did not see any weight change in our study. It was just these A1C and glucose changes. So, yeah, I basically, the team is continuing to work on this, and it would be awesome if we had something that would help with weight loss, for sure. All right, I'm just going to wrap up then, because I think I just wanted to share that when I started this company, there were two, I think, really good papers that existed. One was that fecal microbiome transplant paper. One was this correlative study looking at people with type 2 diabetes versus healthy. I just did a quick PubMed search. There are almost 3,000 papers linking the pathway that we're in to diabetes. And so it's been really cool to kind of be a part of this company where we had these early insights and developing this thing, and in the meantime having all this external academic publications that are supporting that this pathway is really important. And I think you're going to hear a lot more about butyrate and microbiome and all of that as time goes on. And then I'll just end with where I began on this, which the one in the middle is mine. As I said, Annabella is 12 now, and healthy for all intents and purposes. But I'll just share this funny story with you, which is that obviously I've had her on our IBS pills, and we have them in our fridge, and she'd been taking them. And then at one point, I think I forgot to reorder them, and I wasn't even sure if she was taking them or not. And she went to the fridge and she said, "Mom, where are the pills?" And I was like, "What pills?" She's like, "The pills, your experiment pills." I was like, okay. Don't tell other people you're taking mommy's experiment pills. And I was like, "Oh, I haven't gotten more." And she said, "Well, I wish you told me this because we're all going to Baskin Robbins after school." And I was like, "What's that have to do with anything?" And she said, "Well, when I take the pills, I can actually eat ice cream. And so I've been going to Baskin Robbins after school, but only when I'm on the pills." So then we had to have a separate talk about how these aren't ice cream enablers, but actually, yes. But it is very cool to get to create something that you get to see the direct health benefits of. So, yes.
So those pills that you had before, can those just be taken by anybody just to kind of bolster your gut?
So these
just for people who have diabetes?
So these diabetes pills, I actually take them because they help you regulate your sugar. So I enrolled myself in my own trial. There had to be some perks to being in this company. So I wore one of those CGMs, and I did my own placebo. I participated in this placebo-controlled trial, not the one that I shared with you guys, but a separate one. And I actually found that for me, it didn't just reduce my glucose spikes going up, but it also reduced glucose crashes. So when you look at this continuous glucose monitor data, after you have sugar, you get a spike, and then it goes kind of below, and these are, we experience them, right? We experience sugar highs and sugar crashes. And so when we took the data across placebo versus on formulation, the whole thing was minimized. And so there are just basically fewer, it's just helping me to manage my sugar better. So I take them because I'm trying to manage sugar.
So those could be taken by any of us?
They can be taken by anybody, yeah.
So they're over the counter, is that what you're saying?
They are, you can, they're,
The IBS is the friends and family side, but the sugar ones,
we literally released it on Monday as a consumer product. So we have an e-commerce site where we're selling the product. The site is designed to be for people with diabetes, but me personally, I take them for sugar management.
So if we go on to your company website?
You can get a link to that, yes.